much. Thanks a lot for having me invited to speak today. It's a great honor and pleasure. In fact, the title of my presentation only covers half of what I will talk about, because I also like to talk about a bit about the causes of biodiversity. So why do we observe that many species in the environment, which is a classical question, I think, in ecology, and which is needed to understand in the context of experimental studies on biodiversity and also monitoring efforts. So to start, some causes of biodiversity, some in bold because I only touch those that I think are most relevant in the context of this talk. Obviously there are many more. The classical question in community ecology is, how come we have that diversity of species in the environment? The textbook answer to that is, well, there are niche differences, which means that different species occupy different niches and thereby they escape too strong competition with each other, which allows them to coexist. And this is a recent example published in 2004 already of two cyanobacteria, and they have different pigments. And because they have different pigments, they are able to partition the white light spectrum in two, in fact. So one is mostly absorbing the red colors and the other is mostly absorbing the green colors. And so what you see here is nothing but just their absorption spectrum, which is reflecting their different pigmentation. If you grow these two species together in white light, the consequence is, because they partition the light spectrum, is that they coexist in a stable way in these bioreactors. If you take away this niche differentiation by only offering them, for example, red light, then what you see is that the green would exclude the red one. The opposite would happen if you would only offer them green light. The key word here, I guess, is traits. So these are two species that have different traits, and because of that they are able to exploit a different part of the light spectrum and they are able to coexist. However, niche differentiation is only part of the answer to the question why there are so many species on Earth. In fact, there's a second factor that's very often forgotten, and this is what we call fitness equivalence. I will explain what I mean by this. Niche differences I already explained. Example could be light preferences. Fitness equivalence could be the degree to which, for example, fecundity is the same among species. And so these two factors determine if a species could coexist or not. These are theoretical expectations for a very simple competition model, and in the gray zone, species would be able to coexist. In the white zone, there would be one of the two excluding the other. And this typically happens when niche differences are small and or the fitness equivalence is really low. And when niche differences are small, that's easy to understand. When fitness equivalence is low, it means that that two species have very different fitnesses. So you have one that has very high fecundity and the other has a very low fecundity. Just to show that basically there are two factors that influence coexistence, and traits can influence these two factors. So it's not because you have bigger trait diversity that you will also have automatically a much easier species coexistence or a higher diversity. We have shown this for the case of pigments. We are now working on a paper showing that pigment richness does not always promote algal species richness. How do we do this? Well, we have first looked into the literature, and for basically all different pigments that one could find, this is only 6 of them, we found 22 pigments. And then we made a light competition model, so it's a theoretical model, and we parameterized this with these pigments. And what we could do is choosing the number of pigments we put into the model. Either you compose a community only of two pigments, or you would compose a community with all 22 pigments and all values in between. And then we simulated coexistence in the long term. So we tested how many species could survive in the end as a function of the number of pigments in the community. And so what we got was, on the x-axis here, the initial pigment richness, on the y-axis, the final species richness. And what we found was that the most likely case which are the most red colors, and let's say up until yellow, is basically that you only have one species existing despite putting more than 20 pigments in a community. So this shows that pigment diversity actually does very little to help species diversity. So there must be other factors explaining why we have such a big diversity in the phytoplankton, which I mean are also described in the literature, and I will not go into detail here. But the point is that trait diversity does not automatically mean you have a high species diversity. When we move from the local to the regional scale, then most people know that environmental heterogeneity is a key driver. Let me explain this a bit. So if you have a very homogeneous landscape, and here all the different dots are local communities, and the lines between those communities are connections, and that allow individuals to move from one local community to the other. When the environment is very homogeneous, then it means that there are not really different habitats. There's just one habitat spread across the whole landscape. When the environment is really heterogeneous, then you have different environments. So local communities have different environments. This allows the creation of different habitats, if you want. For example, here the red dots could be local communities with a high temperature, and the blue ones could be local communities with a low temperature. And theory would say that in case the landscape is really homogeneous, you have a low diversity, because you have the same habitat everywhere. You would get the same species specialized on that habitat everywhere. So regionally, you have a low diversity. When the landscape is really heterogeneous, then you would have maybe some low temperature adapted species in the blue patches, and then the temperature, those that prefer high temperature in the red patches. So you would have a higher regional diversity when you have a heterogeneous landscape. That's quite well known. What is not so well known is what the influence is of the connectivity between these different local communities. And this is something we have tested using microcosm experiments with algae. So we have our different Erlenmeyer flasks with our experimental communities, and we connect them via pipetting, and then we also install a treatment of heterogeneity by treating some of these local communities, in this case with a pesticide, and leaving others untreated. While in another treatment we would have all patches untreated to create a homogeneous landscape. And what we found was that first, the heterogeneous landscape have a higher regional diversity than a homogeneous landscape, which is what we already knew or could suspect before. But what was interesting was that in fact the degree of connectivity also mattered a lot, because more connected landscapes, for more connected landscapes, the difference between heterogeneous and homogeneous was much bigger than when the connectivity was really low. And the reason for this is quite logical. If connectivity is really high, then it gets much easier for species to find their optimal habitat. So the effect of the heterogeneity on regional diversity gets magnified by this connectivity. This also sounds very theoretical, well, in fact it is, but I am also convinced that there is an application possible to do field studies, simply because when we look to water currents, then it's clear that the sea is not a well-mixed bioreactor. So some parts of the ocean are probably more isolated than other parts. And on top of that, the environmental conditions are also going to change. So environmental heterogeneity or homogeneity is a factor that is also varying out there. Plus also the connectivity is not 100%. So there are some patches that are more isolated than others. And even more so, if we look at the benthos, then bioturbators are known to have completely changed the structure of the subsurface, so the sea floor, if you want. But also importantly, they have increased a lot the spatial heterogeneity. I apologize for the two terrestrial species on this slide. She or he who can detect them, I probably should buy a beer afterwards, so I'll quickly go continue. Okay, so enough about the causes of biodiversity, now about some consequences, so the effects of biodiversity. When we talk about the effects of biodiversity, well, typically what people know, if they have heard about it, is the case of the grasslands. So very far away from what we are talking about today, I realize, but let me come to my case afterwards. This is the type of result that people obtain. So in a grassland, if you put more species together, then you would find a bigger productivity. And this result is quite consistent and even gets stronger through time, apparently. However, this is not necessarily the case in other systems, and notably in marine systems. What we found with some of our work is that in these coastal diatom communities, the productivity actually becomes lower when you put together more species. So why would this be the case? Well, the main difference is that in the grasslands, complementarity, positive complementarity, is a key effect of biodiversity on function. The more you put species together, the more likely it becomes that these species will have different traits, here for example rooting depths, that allow them to maximize the amount of nutrients they can take up and convert into biomass. If you compare this to our simple plankton systems, which are well shaken and probably represent the very local conditions in some parts of the ocean, there the spatial heterogeneity is almost absent. And in that case, what you get is so-called negative dominance effect. This means that you have one or a few species that are very, very well at capturing the resources, and as such outcompete the others. Now, unfortunately, apparently, these species that are very well at capturing resources do very little with it. They grow not so much, so they, in fact, they produce little biomass for the resources they capture. So you get a negative dominance effect. And the more species you put together, again, the more likely it becomes that you have one of these nasty species in your community. That's why you get a negative effect of diversity on function. To make things even a bit more complicated, biodiversity effects on function do not only change among systems, they also change with the environmental condition. This is a paper showing that temperature treatment is going to change the effect of biodiversity of the x-axis on function, here total absorption as a proxy for biomass, going from, let's say, moderately positive to a bit more positive in the middle temperatures to a flat line at the highest temperatures. So to synthesize, this is more or less what they found. So the slope of the BEF, so the biodiversity because the function relationship, first goes up, reaches a peak, and then goes down. What we hypothesized in our recent paper that will appear, I guess, next month or so, is that this trend is a quite general one. Why? Because the mechanisms behind it are really, can be found back in many different systems. What are these mechanisms? The first mechanism is the one that causes the initial increase, so the left part of this unimodal response. And this is nothing else but a so-called richness-dependent buffering. It means that richness buffers harshness. What do I mean by that? If we look at the blue arrows, you can kind of quantify the response of the monocultures, so the systems with only one species, to a shift in temperature. And there is quite pronounced effect, I should say. If you make the same exercise for the highest diversity levels, so those that contain 64 species, the effect on the production is much smaller. And this is the classical example of the portfolio effect. So the more you have species, the more you have compensations. When one species is sensitive to temperature, another species would compensate for that. So the more species you have, the bigger this buffering effect gets. And this buffering effect makes this line always shift up. No matter how, it always shifts up, because it is pushed down here more than it is there. Afterwards, from this maximum onwards, at some point it gets too hot for everyone. It gets too harsh for everyone. So then you get a very low function, despite the initial number of species you have put in your community. And so the BF slope would fall back to a flat line. This unimodal response, we tested it using a meta-analysis, and we collected more than 50 studies. I admit mostly grassland studies because this is what the most data were, well, this is where we found most data on. There are also some marine studies, but those are the studies we did ourselves. And there are two clusters here. There's the red cluster, the blue cluster. The red cluster obeys the unimodal response. The blue cluster doesn't. I won't go into detail why, we don't have the time. The point here is that we could predict using very simple competition models both clusters. So we could predict the unimodal response, and we could also predict the cases where the response is monotonous, decreasing, so the blue category. Okay, again, seems quite theoretical, but there is a quite important implication, I would say, because if you put on this slide to the BEF slope versus harshness, you could simply replace BEF slope by importance of biodiversity, because if the slope is big, then it means that losing a species has big consequences for function. If the slope is flat, then losing a species has smaller consequences for function. And harshness you could change by environmental change. And now, if I am a bit optimistic, maybe, and I think that maybe at the present we are more or less here, so there is environmental change going on, but not yet up to a critical point where communities start falling back to a flat line and nothing is functioning anymore. Okay, so let's say we're more or less here. Then any increase in environmental change is gonna make the importance of biodiversity bigger. So this means that we predict that biodiversity protection will even be more important in a world that is changing, exactly because of this buffering effect. So it will be key to have a sufficient number of species to buffer ecosystems against future change. This is what this slide is showing. So more biodiversity protection, yes, but let's not focus too much on richness, the number of species, even though this is the classical indicator used in many studies. This is why, because this is what's going on at the moment. This is a study from a couple of years ago. This is the number of species that you would find back in the local community. All these different lines are local communities, and the colors tell you where the data come from. And this is the overall trend for the planet, basically. Locally, there is not a consistent loss or gain of species, locally. But this, however, has some different stories. This is a so-called Jacquard index, and this tells you to what extent there is temporal turnover. And the fact that this line is decreasing means that locally, communities are undergoing change. So they are swapping species. So that it's like they lose a species, but they immediately gain a new species, such that the number of species stays the same, but the composition changes. And this is what is happening actually locally. So this shows that we should not focus too much on richness, but also on things like composition. When you talk about biodiversity, it gets also very easy to forget about the very simple, down-to-earth things, like, for example, density. We have calculated that it's very important to also keep the population size in check for ecosystem function. Very simple. You have two sites A and site B, and you want to compare the function between these two sites, and they only differ in density. They do not differ in richness. They do not differ in composition. What is the maximum difference in function you could get between these two sites? And I present some results in the next slide by dividing by the function at the reference site, just so I have a proportion. It's easier to compare. These are some quantifications. So let's now only focus on the y-axis. So this is the proportional change in function. So remember, two sites, the only thing they differ in is density. The same richness, the same composition, same species present everywhere. Still, you could find differences between plus 40% and minus 30%, depending on which site is the reference site, basically. That's not so important now. The main point here is that these differences can really be substantial. So you can have a monitoring data set of site A and site B, say, oh, we're very happy because our site near the our site near the harbor has the same number of species and the same kind of species as our reference site. So we're very happy. Functioning must be okay. The ecosystem must be okay. Well, no, because both can be radically different in terms of function. What are the different categories that I plot here? Well, these are technically covariances. We won't go into detail there. The point is they characterize the identity of the species that suffer most density depression going from one side to the other. So they put information into the analysis of which species are depressed most, without losing them, but just depressed most in terms of their biomass. And again, here traits matter, of course, because if you're gonna depress mostly the density of species that contribute a lot to function, then the difference between the two sides in terms of their function is going to be bigger, of course. This echoes, but also quantifies, points that have been raised a couple of years before in this paper, saying that mass extinction is important, but mass rarity could have contributed much more to the biogeochemical cycles at geological timescales than the loss or gain of species. So after a perturbation, sure, you find back many species are extinct, but the density of individuals is also much, much lower here than there. And this loss in density, the fact that you have much smaller population size, could be much more important for ecosystem processes than the loss of species. And then to finalize, I have two short ideas that I just want to introduce that I think are interesting for future research directions. The first point is that I have now always, and together with me many others, focused on communities composed of one trophic level, in my case phytoplankton. But the phytoplankton, of course, is only a part of what are in reality quite complex food webs, as we call them. And these food webs even include human activity, for example, by fishing. So how to define diversity for such complex food webs, and what is the influence of that kind of diversity for ecosystems? There is a recent paper out showing that vertical diversity, so it's the number of trophic levels you would find back in a food web, has a big effect on function. In the same way that very often in grasslands people find more species to produce more biomass. And then the last point is that this I think I won't present. The last point is that we need to better integrate biodiversity across levels of organization. To be more precise, we need to understand how diversity within species is important to sustain or make more harder the coexistence among species. And to finalize, this is a new project that is now starting, where we want to test if or when intraspecific variability matters for species coexistence. And the project is a kind of distributed experiment, so we are various partners, and we are still actively seeking for laboratories to adopt the protocol that we have developed, such that we have such that we can replicate our experiments across a broad range of laboratories for different study systems and obtain a much more general answer to the question whether or not intraspecific variability matters for species diversity. Thank you.